Hello everyone and welcome to today's webinar, Immunity Comes First, a Sustainable Ally from Algae. My name is Nick Taylor and today we'll be discussing consumer insights on immune health and how a specific alga, Euglena gracilis, possesses the ideal composition of essential nutrients and bioactive ingredients that could sustainably provide immune support. Our speakers today are Mike Hughes, Director of Insights at FMCG Gurus, Philip Calder, Professor of Nutritional Immunology at the University of Southampton, Jeff Horst, R&D Lead Beta-Glucans at Kemin, and Samantha Machi, Senior Commercial and Technical Manager for EMEA and LATAM at Kemin. Just before we get started, I have some housekeeping items to go over. If you have any questions for our panel, please type them in the Q&A box below and hit the submit button. We will answer as many questions as time allows. Okay, with that cleared up, I would like to hand off to our first speaker. Mike has over 13 years experience analysing consumer trends and has a particular interest in challenging industry perceptions about how consumers think and behave and identifying new trends across markets. Mike, you have the floor.
Hi, my name is Mike Hughes and I am Head of Research and Insight at FMCG Gurus. And what I'm going to do now is give you a brief overview of consumer attitudes towards immune health, understanding how consumer attitudes towards their immunity are evolving, why consumers deem immune health to be important, and what steps they're looking to take to address their immune health. So across the globe, consumers are not satisfied with their immune health. In Q3 2019, FMCG Gurus surveyed 26,000 consumers across the globe in respect to their attitudes towards their immune health. The research found that only 54% of consumers said that they were currently satisfied with their immunity. This showed that a considerable proportion of consumers currently feel that their diets could be improved, that their general lifestyles could be improved, and that they are somewhat vulnerable to illness. Fast forward 12 months, and this figure has dropped to 49%, showing only 1 in 2 consumers show satisfaction with their immune health. At the same time, there has been a significant increase year on year in the proportion of consumers who are saying that they are making changes to their diets and lifestyles in order to improve their immune health, with 70% saying that this is the case in 2020. This shows how consumers are taking a long-term and proactive approach to health as they look to minimize the risk of disease and illness. Consumers associate a poor immune system with a variety of negative health attributes. Ultimately, consumers associate a poor immune system with being easily susceptible to long-term health problems. Across the globe, we know that consumers are adopting the notion of healthy aging, wanting to stay fit and active until late as life as possible. At the same time, they recognize that vulnerability to illness is something that increases as they get older. As such, as consumers look to break stereotypes associated with older age, they will look to maintain their health and lead an energetic and vibrant lifestyle. Additionally, consumers also associate a poor immune system with high levels of fatigue. And I think that's something to point out in today's modern society, with many consumers often feeling tired, stressed, fatigued as they cram as many activities into the day as possible. And many consumers are concerned that this is something that can not just impact on their mental well-being, but actually that's something that can also impact on their overall immune system, making them vulnerable to disease and illness. Now, across the globe, we know that consumers are adopting the notion of holistic health. This is where consumers recognize that all aspects of health are interlinked and connect with each other and should not be treated in isolation. As such, when consumers look to make plans to improve their immune health, they are looking to improve other aspects of their health as a result of this. FMCG Gurus research shows that across the globe, a significant proportion of consumers believe that by improving their immune health, they will also improve their cognitive health and also their digestive health. We know that these are two issues that have become more common over the last couple of years. For instance, across the globe, a higher proportion of consumers are saying that they are suffering from problems such as constipation, gas, bloating, and other stomach pains, which not only cause discomfort, but can also significantly impact quality of life. At the same time, consumers are associating good digestive health with being central to good overall health. And as mentioned, consumers are not satisfied with their cognitive health. They have a variety of daily stressors, which can impact on their ability to relax and unwind at night, to have a good night's sleep pattern. And this is something that can often lead them feeling tired, fatigued, and void of energy throughout the day. Something that's very recognized can impact on their physical health as well as their cognitive health. As a result of this, as consumers take a more proactive approach to addressing their immune health, they are also hoping for wider benefits, such as improved digestive and cognitive health. FMCG gurus asked for 7 in 10 consumers who were planning to improve their immune health what steps they are taking. The research shows that the top 3 steps being taken by consumers are to make changes to their diets, to improve their energy levels, and to exercise more. The importance of exercise shows that consumers are taking a holistic approach to health, whilst the importance of energy levels further highlights the importance of consumers not feeling fatigued. However, consumers recognize that their diets are not as healthy as they could be, something that will lead to health problems later in life. When asked what steps they will be taking to improve their diets, consumers highlighted a 2-pronged strategy. Firstly, they will reduce their intake of ingredients such as sugar and additives. These ingredients are associated with being dietary evils that offer little to no nutritional value and are seen as something that contributes to health problems such as obesity and diabetes. At the other end of the spectrum, consumers want to increase their intake of food and drink that has genuine nutritional value, which means they will turn to more fresh fruits and vegetables and also look to increase their intake of protein and vitamins and minerals. Consumers seeking out ingredients that offer a health benefit beyond basic nutrition. As mentioned, concerns about immunity relate to consumers adopting a long-term approach to health. We asked consumers why did they want to make changes to their immune systems. The research showed that the most popular answer was that consumers were concerned about developing long-term health problems. This shows that ultimately consumers recognize that their current diets and lifestyles are not as healthy as they could be, something that could lead to health problems later in life. At the same time, and linked to this, consumers were concerned about developing disease and illness and want to maintain a good quality of life. And this really shows that when it comes to analysing attitudes and behaviours towards immune health, consumers are adopting a long-term and proactive approach to addressing their immunity so that they can stay fit and active until as late as life and possible. That concludes my presentation. Thank you very much.
Thank you, Mike, for that very interesting presentation. As a reminder, if you have any questions for our panel, please type them in the Q&A box below. We will now move to Professor Philip Calder from the University of Southampton. Philip is an internationally recognised researcher in nutritional immunology with numerous publications and many conference presentations. He is currently president of the Federation of European Nutrition Societies. Over to you, Philip.
Thanks very much for the kind introduction. Welcome, everybody. Today I'm going to talk to you about the immune system and a special role for beta-glucans in supporting immunity. My talk will cover these points. I want to introduce the immune system to you with a simple overview, say something about how the immune system changes over the life course, the fact that it varies between different people and what the sources of that variation might be. Then in the second part, move on to talk about beta-glucans, immunity and infection, then discuss with you why beta-1,3 glucans are really special in supporting the immune system, and then I'll summarize to wrap up for you. So the immune system is a cell and tissue system that helps people protect themselves against pathogens, bacteria, viruses, and so on. We know that a well-functioning immune system is key to providing good defense against these pathogens. And we know that because people who have weakened immune systems or who are immunocompromised are at much greater risk of becoming infected and at much greater risk of infections being more severe. Essentially, the way the immune system works is it distinguishes things that belong from things that don't belong, what the immunologists have called self and non-self. The four general functional features of the immune system are listed here. Firstly, it acts as an exclusion barrier to keep pathogens out. Secondly, it identifies or recognizes pathogens if they break through the exclusion barrier. And this process of recognition is key to my talk and is key to the way that beta-glucans influence the immune system. Thirdly, having identified pathogens, the immune system acts to eliminate them. That is, it destroys bacteria and it destroys virally infected cells. Finally, is the memory component. So the immune system actually develops a memory of its encounters so that if individuals become reinfected, the response is much quicker and more vigorous. This memory response of the immune system is, of course, the basis of vaccination. So I mentioned these different levels of immune activity. In general, the immune system is classified into two components, the so-called innate or natural immune system and the acquired or adaptive immune system. The components of those two parts of the system are listed here. So we see the physical barriers are part of innate immunity, but there are also cellular and protein components of innate immunity. So, for example, phagocytic cells like neutrophils, monocytes, macrophages, and dendritic cells, these are parts of innate immunity, as are natural killer cells. Inflammation is part of innate immunity. Then adaptive immunity involves lymphocytes, T lymphocytes or T cells, and B lymphocytes or B cells. These form the cell-mediated immune response, and B cells are responsible for the production of antibodies and what's called humoral immunity. The memory response involves T cells and B cells, so that's part of adaptive immunity. So we have these two general classifications, innate and adaptive, but in fact, they communicate with one another. They're linked. So as a simple example, phagocytic cells like dendritic cells engulf bacteria and kill them intracellularly. Then components of those bacteria are expressed on the surface of the dendritic cells, and they are recognized by T lymphocytes. So this is a direct interaction between the innate immune system and the adaptive immune system. The T lymphocytes then become active and control the activity of other immune responses, for example, antibody production by B cells. So the key to all of this immune activity is the recognition of invaders. So the immune system has to be able to recognize thousands, maybe tens of thousands of different types of bacteria, viruses, and so on. So to do that, it has developed a series of receptors which are called pattern recognition receptors. These receptors do exactly what their name suggests. That is, they recognize patterns on the surface of bacteria or virally infected cells. They also recognize bacterial components that have fallen off of bacteria, for example. So the things they recognize are called microbe-associated molecular patterns. The best described of the pattern recognition receptors are the two on the left-hand side, the toll-like receptors. These recognize different bacterial proteins. You'll see next to the toll-like receptors, there's a pattern recognition receptor called dectin-1 that recognizes beta-glucans, and I'm going to come back to dectin-1 later on in my talk. So far, I haven't mentioned the intestinal immune system. So within the gut wall, immune cells are compartmentalized in particular structures. For example, in this diagram, we see on the right-hand side Peyer's patches, and on the left-hand side, lamina propria. This is where immune cells congregate and communicate with one another. But the main role of this gut-associated immune system is sampling things that are in the gut lumen. This could be sampling the bacteria that are present, or it could be sampling the remnants of food that are present. This is important because it means the contents of the gut are sampled by the immune system, and signals are taken back by the immune system, for example, to lymph nodes, shown in the lower right-hand side here, where the immune cells make decisions about what's been sampled. So they make decisions about the gut bacteria, they make decisions about the food remnants, and so on. Having made those decisions, some immune cells can move back to the gut wall, but importantly, some immune cells move elsewhere in the body, for example, to the lungs. And what this means is that there's an immunological connection between the gut and between the lungs. So the contents of the gut can actually affect the immune system in the respiratory system and elsewhere. It's important to keep in mind that the immune system changes across the life course. So babies are born with an immature immune system that becomes mature or educated, as I call it on this figure here, over the first weeks to months to years of life. And there are particular factors involved in that immune education. Later in life, the immune system declines. This is called immunosenescence. There's a decrease in output of new immune cells from the bone marrow and from the thymus, and immune cells from the blood have weakened activity. That means older people have weak immune systems. In this figure, I hypothesize a threshold below which the immune system is not sufficient to give people protection against pathogens, so the individuals would be at increased risk of infections. This also results in poor responses to vaccinations in people receiving vaccines. So there's this general pattern across the life course of early maturation and later decline. But if we look across many different people, we see different patterns. So, for example, in some infants, the maturation can occur faster, in others slower. In some older people, the decline can occur earlier and can be faster, or in fact, it may not even be apparent. So there are many older people who still have very robust immune systems, for example. And if we look in middle-aged people, we can see they can be at different levels of immune competence. And the part that looks a bit like an upside-down triangle is trying to capture the idea that within one individual, the immune system can change for a period of time and then maybe can revert back to another position. So what is the source of this variation in the immune response across the life course and between individuals? Here I capture some of the factors that impact on the immune system, giving rise to this variation. So it's obvious that being infected or being vaccinated could impact on the immune response, but also people's genetics, whether they're ill, the medications they take. At the bottom of the figure, lifestyle factors like smoking, alcohol use, physical activity can all influence the immune response. I mentioned aging, but also the immune system changes during pregnancy, for example. It's influenced by obesity, by the gut bacteria, the microbiome, and also by nutrient intake. And as we heard from Mike, stress also impacts the immune response, both psychological and physiological stress. So there are many factors that can influence the immune response, and these factors, of course, are different between people, but they can also change over the life course. So having given you a feel for the immune system, its components, and how it's different between people, let's now look at beta-glucans and immunity. So I think it's interesting to draw your attention to two reviews that have been published this year. I was involved in the first of these reviews, published in Molecular Nutrition and Food Research, which is focusing on beta-1316 glucans and immunity. And then there's another review published in Frontiers in Immunology, looking at structure-function relationships of beta-glucans in the context of immunity. So these two up-to-date reviews are really providing a wealth of information on this topic of beta-glucans and immunity. Now, if we go back to the importance of recognition within the immune system, I mentioned that one of the pattern recognition receptors, dectin-1, recognizes beta-glucans. And this is the key to the ability of beta-glucans to support the immune system, maybe to promote immune activity. Dectin-1 is an interesting structure. Here the dectin structure is indicated, so it has this head poking out of the cell membrane which can bind beta-glucans, and that's linked to an intracellular domain which is involved in signaling. So that when beta-glucans bind to dectin-1, two general types of response are generated. I've circled these in red. On the left is what we call an antimicrobial response. This is the promotion of phagocytosis, which is the engulfing of whole bacteria and their digestion, and the generation of an oxidative burst. This serves to kill the bacteria through generation of oxidative stress within a vacuole inside the cell. So in other words, beta-glucans acting through dectin-1 can trigger antimicrobial immunity. The second red circle captures the other parts of the signaling from dectin-1. First of all, generation of an inflammatory response, and then the generation of antigen presentation to initiate the adaptive immune response through T cells and B cells. So dectin-1 is behaving within innate immunity. It's helping with the phagocytosis and the destruction of microbes, but it's also linking innate immunity with the adaptive immune response. Dectin-1 is expressed on monocytes, macrophages, dendritic cells, and neutrophils. These are all phagocytic cells. It's also expressed on some immune cells called M cells within the gut-associated lymphoid tissue. And I mentioned before the importance of the gut-associated lymphoid tissue in our ability to deal with gut bacteria and also food remnants. It turns out there's actually a second receptor in the immune system that interacts with beta-glucans, and this is a receptor called complement receptor 3, or CR3. This figure depicts inactive complement receptor 3 on the left and the active receptor on the right. So you'll see the receptor has two different subunits, and when it's activated, the way these subunits interact with one another changes. Now, complement receptor 3 has two binding sites. One of them is shown at the top of the figure, and this is the recognition of bacteria which are coated with a protein called complement. This process of coating is called opsonization. So the complement receptor recognizes these coated bacteria. So this is a signal to the immune system that bacteria are present. But another signal is needed for the complement receptor to become activated, and that's shown at the bottom left here. It turns out that the complement receptor 3 also has a domain for binding beta-glucans. And it turns out that complement receptor 3 works best when beta-glucans are bound and the opsonized bacteria are bound. So this is what's called the dual ligation. So you might imagine that beta-glucans could be bound to the complement receptor already, and it becomes active when bacteria are present and are opsonized by complement. This will drive the activation of complement receptor 3, and you'll see the effects of that are on the right-hand side. Again, phagocytosis, destruction of bacteria through reactive oxygen species, and the generation of an inflammatory response. So we see these two receptors, dectin-1 and complement receptor 3, that both bind and respond to beta-glucans. So you might imagine beta-glucans can affect the immune response. So this is a table taken from one of these reviews, listing the different, mainly cell culture, but also some animal studies that have looked at the ability of beta-glucans to promote immune responses. I won't go through the details, but you'll see in the column called immunomodulatory activity, there are a lot of arrows going up, indicating that exposure to the beta-glucans enhances many different components of the immune response. Now, this seems to occur in this table at least, summarizing data from 1316 glucans, the beta-1,6 glucans, and the beta-1,3 glucans. So this could be a general property of beta-glucans. But what's important to note is that it's actually the beta-1,3 glucan that is what is recognized by both dectin-1 and by complement receptor 3. So in other words, the important glucans in driving the immune response seem to be the beta-1,3 glucans. And here this figure from our review is just linking the effects through dectin-1 and complement receptor 3 to phagocytosis and destruction of bacteria within that vacuole that you can see. There's a mouse study which has looked in some detail at how beta-1,3 glucans work. So it turns out in this study it was shown that oral beta-1,3 glucans are taken up by immune cells, particularly macrophages, within those structures in the gut wall that I talked about before, the Peyer's patches. Once the macrophages take up the beta-1,3 glucans, they actually move elsewhere in the body. I talked about that before. They move to the spleen, to lymph nodes, and to the bone marrow. Beta-glucans, of course, can be large structures. The macrophages can degrade the large beta-glucans into smaller soluble beta-1,3 glucan fragments, and it's those soluble fragments that bind to the complement receptor 3. So it seems that dectin-1 could be a way of sampling beta-1,3 glucans, and then there's this link between the degraded beta-1,3 glucans and the binding to complement receptor 3 on neutrophils now. So this gives rise to the idea that beta-1,3 glucans are actually priming the immune system. I mentioned this before, that soluble beta-1,3 glucan could be already bound to complement receptor 3 prior to bacterial exposure. So this is sort of setting the immune system to respond once bacteria are recognized. So this is the idea of immune priming, which seems to be a very important aspect of how beta-1,3 glucans work. So is there evidence that beta-1,3 glucans, okay, they affect the immune system, but can they have an effect on infection? So here's a mouse study using the algae Euglena gracilis that contains beta-1,3 glucans. And this also looked at the algal structure where these glucans are stored. So it's a mouse feeding study involving the algae itself, the beta-glucan storage structure from the algae compared to control animals. So these mice were fed either a control diet, a diet containing the algae, or a diet containing this beta-glucan rich structure. And then they were exposed to influenza, the influenza virus. And you'll see that over 6 days, most of the control animals died, over 80% of them died. But there was actually a survival advantage if the animals had been pre-fed the algae or the beta-glucan containing algal structure. So beta-glucans perhaps enhance survival to a viral infection. This could be because of their enhancement of immunity, their immune supporting and their immune priming activities. So I think that's actually how the algae is working in this experiment, because what the researchers also did was they looked at how much virus was present in the lungs of those animals. So all of these animals were infected with the same amount of virus, but if the immune system is working, they would have less virus present. So you see the viral numbers in the lungs of the control animals peak at week 2 and then go down a bit as the immune system kicks in. But you'll see the viral numbers are always lower in the other 2 groups which are receiving beta-glucans. So I think this is an indication that the algae and its beta-glucan containing structure is helping the immune system deal with the virus to which these mice have been exposed. And that's the explanation for the better survival. So what I've told you is the immune system is important to a healthy life. It's actually a matter of life or death, in fact. It's complex. It involves many different cell types, lots of chemical mediators, lots of interactions. It has mechanisms for recognizing non-self structures, including beta-glucans. It develops early in life and declines with aging. There's variation between immunity between people, and I talked about the many different sources of this. Then I went on to mention that there are 2 receptors within the immune system that recognize beta-glucans, dectin-1 and complement receptor 3. There's quite a large literature on beta-glucans, immunity and infection, involving cell culture, animal and human studies. This is mainly about the 1613 glucans. However, it seems that beta-1,3-glucan is the key structure involved in the immune effects of beta-glucans, including this priming effect. What's clear is I think we need more human trials of beta-1,3-glucan, and we need to think about the target groups of those trials. Thanks very much for your attention.
Thank you, Philip, for that insightful presentation. Our next speaker is Jeff Horst, a marine biologist by training and co-founder of the world's first commercial scale producer of beta-glucan from algae. Jeff, take it away.
Thank you, Nick, for the kind intro. So my goal today is to give you the brief history about BetaVia and how we came about developing this algae-based beta-glucan product. So as Nick mentioned, my background is in marine biology, and a little over 10 years ago, I was one of the co-founders for a company called Algal Scientific, and we developed a technology to use algae to treat the wastewater from the food and beverage industry. So imagine all the nitrogen and phosphorus that's involved in making beer. It turns out algae are really good at soaking up those nutrients. And one of the species of algae we were using was Euglena. And if you were to search the internet for info about Euglena, you'd realize it's one of the few species of the world that produces beta-1,3-glucan. And so Dr. Calder just gave a really nice background about how that very specific molecule is a really potent immune modulating molecule. And so we figured maybe we could utilize our expertise in growing that algae in wastewater and maybe apply it to growing Euglena under highly controlled conditions to make a nutraceutical product. And so we spent about 18 months to really break all the records as far as growing this algae under really dense conditions, under fast growth rates, and maximizing beta-glucan, and importantly, scaling it up to industrial scale. So many researchers have been growing Euglena in the lab for decades, but nobody had been successful at really growing it at the industrial scale, which is required to have a economically viable product and process. And so just to give you kind of a picture of what this looks like, the top picture on this slide shows you two Euglena cells that are in the process of dividing. And you can see inside there's these kind of marble-like structures. Those are actually the beta-glucan granules that the algae make to store energy, just like how some algae store energy as oils and plants store energy as starch. This algae stores extra energy as beta-glucan. And so while we were maximizing the growth conditions for growing Euglena in sterile fermentation tanks, we also needed to really establish the science about how our form of beta-glucan compared to the other beta-glucans that are available from other species that are not algae. And so we funded studies ranging from the cellular level studies that Dr. Calder was talking about all the way up through animal studies and including human clinical trials. And so my colleague Samantha will explain a little bit about our most recent human clinical trial. But the point was to really demonstrate how our product compared to the other beta-glucans, as well as to understand the dosage rates, and also, of course, demonstrate safety. And so we were able to achieve all of that as a startup, as Algal Scientific, and it was a little over 3 years ago that Kemin came in. They were looking for an immune product, and so they really liked what we had. And so now, after being acquired by Kemin, we've been able to introduce BetaVia not just in the United States, but across the globe. And so I'm happy to still be contributing in that fashion. I'll give you a little bit more information about how this product is made. And so if you were to come to our fermentation facility in Michigan, you would think that you were in a microbrewery. So we grow the algae in these large sterile fermentation tanks, so these stainless steel tanks. And it's a continuous process. So that means that we harvest a portion of the tank every day and send that algae culture to a process that concentrates the algae cells into a paste. That paste is then dried, and then we mill those dried flakes into a powder. And so just like that picture there, BetaVia Complete is really just dried and ground up Euglena gracilis cells. So there's no extra chemical ingredients. We don't have to use any chemical extractions at all. There's a lot of advantages for doing sterile fermentation. One is that we can control contamination in those tanks, so we can keep out any unwanted microbial contaminants. We also have complete control over all the ingredients, so that allows us to have really high quality control of what's going in. So compared to an outdoor pond where you really can't control the birds and other things that might be dropping into those ponds, we can have complete control. We also have very good batch-to-batch variability because it's really the same culture that's being harvested every day. So the material we make today is the same as on Saturday, and it's the same as what we're going to produce in December. So there's also some environmental benefits to growing the algae in fermentation tanks, and the most notable is really water savings. And so by growing the algae in these dense fermentation tanks, we use about 5 to 10 times less water than what would be required if you were growing these in outdoor ponds. And that actually translates into substantial energy savings as well, because it takes a lot of energy to concentrate those algae cells into a paste and then finally to dry it. And so for Kemin, we track the environmental impact of all the ingredients that we make, and BetaVia is actually one of the better ingredients in that regard because of these substantial energy and water savings. I already mentioned that we don't use any harsh chemical extracts, and a lot of people wonder if it's a GMO strain, and the answer is no. So while we've had this culture in our labs for going on 10 years now, and it's adapted to our culture conditions, it is not a GMO strain. So just to circle back to what BetaVia Complete is, it's a dried Euglena gracilis fermentate. We've maximized the amount of beta-glucan in the product, but there are still the other healthy benefits like highly digestible proteins, high mineral content, a small amount of omega-3 fatty acids, and some interesting vitamins and pigments that all contribute to some of the healthy added benefits of the whole algae product. So I'll pass it along now to Samantha, and thank you very much for your time.
Thank you, Jeff, for that informative presentation. Our final speaker is Samantha Machi, a biologist by training and Senior Commercial and Technical Manager for EMEA and LATAM at Kemin. Over to you, Samantha.
Thank you, Nick, and welcome to all the participants. We learned from Professor Calder about the specificity of beta-1,3-glucan and the role they may have in immune response. And my colleague Jeff has guided us through the development of a new and sustainable source of beta-1,3-glucan from algae and has described the characteristics of the whole Euglena gracilis fermentate that we call BetaVia Complete. So in this last part of the webinar, I will share with you the scientific evidence addressing the use of BetaVia Complete in product design for immune support. BetaVia Complete can support immune health by its triple action protection. It primes key cells of the immune system. It's able to fuel the growth of healthy gut bacteria, thus acting potentially in a prebiotic-like manner, and protects the intestinal barrier integrity, something that we learn is important for the exclusion barrier. And these mechanisms are supported by our preclinical research. Focusing on the first of these mechanisms of action, so the ability to prime key immune cell, beta-1,3-glucans from BetaVia Complete reach and digested the intestine. Here are recognized by macrophages and dendritic cells present in the gut-associated lymphoid tissues that can ingest beta-1,3-glucan in a process called phagocytosis. And beta-1,3-glucan fragments, together with the signaling molecule, can then be released, and these steps are important for recruiting additional immune cells. This can result in enhanced phagocytosis of foreign particles and provide overall immune function support. We can see this happening in vitro. So these pictures taken at the electron microscopy show on the left a macrophage indicated by the blue arrow that is engulfing a beta-1,3-glucan from BetaVia Complete, indicated by the arrow, the red arrow, and on the right, the cell, the macrophage cell, that is has already done phagocytosis of 4 granules and is in the process of engulfing a fifth. Then fragment will be released, and this can prime other cells of the immune system. These mechanistic data were supported by a randomized double-blind placebo-controlled trial that was published at the end of last year. It aimed to assess the effect of Euglena gracilis fermentate, so BetaVia Complete, on immune function in healthy active adults. We can see that participants were adult, 25 to 65 years old, overweight up to obese, supplemented during 90 days with BetaVia Complete or placebo, and visit performed at screening baseline 90 days, and then an additional assessment at 30 days. Efficacy was assessed with the Wisconsin Upper Respiratory Symptom Survey, focusing on symptoms of upper respiratory tract infection, URTI. So URTI can cause cold and flu symptoms and are the most common form of illness, acute illness, with 70% of the population that at least once a year experience an episode of cold and flu. And certain stressors, such as intense aerobic exercise, can cause transient immunosuppression and make people more prone to experience these symptoms. So in the study, we selected to include people with performing intense aerobic exercise, 1.5 hours per day at least, 5 days a week. And also participants who had flu vaccination were allowed to participate in the study. This is an example of the Wisconsin Upper Respiratory Symptom Survey. So it's a validated form that asks participants about if they are sick or not, if they have symptoms of cold and flu, classical symptoms, and how these impact their functional outcome. If they are sick or they have symptoms, they have, in addition, to rate the severity of their being sick or symptomatology. And so these enable to assess and to collect a lot of valuable information that can be then processed and provide overall idea of the effect of the product. We saw an effect of BetaVia Complete in many of the outcome assessed with the WURS. I'm starting with the global severity of symptoms. That is a parameter providing the area under curve of frequency, duration, and severity of the symptomatology. And we saw in the chart throughout the various chart in gray, placebo, and in red, BetaVia Complete, that BetaVia Complete supplementation was able to reduce in a statistical significant way the global severity of symptoms at from day 1 to 30, and again day 1 to 90, resulting in 80% lower severity of symptoms. Another interesting parameter was the sick days. Also in this case, we saw a statistical significant reduction at 30 and 90 days compared to placebo, resulting in 3.3 fewer sick days. This is interesting considering that we are talking an healthy population, so a good achievement in 90 days period. Also reduced was the effect on URTI symptoms days, so the number of days with URTI symptoms, trending effect at 30 days, and a statistical significant reduction in 90 days with BetaVia Complete supplementation. At the end, 10 fewer URTI symptom days per person over the 90 days period. Episodes were also positively affected by supplementation. Again, trending effect at 30 days and statistical significant difference at 90 days. So an episode is a sudden appearance of a symptom after at least 2 days in which the person was considering not sick. And we saw a reduction in two fewer URTI episodes corresponding to 45% reduction. The last effect that I will show is the URTI symptoms, so the number of symptoms per person. Statistical significant reduction at 30 and 90 days compared to placebo. Overall, 70% reduction in URTI symptoms. So these represent the overall symptoms, considering all the 13 symptoms, but the WURS questionnaire enable the assessment of each of the symptoms and see how they change during the study. This is what we did in a post-hoc analysis. So in this color-coded bar, you can see placebo, symptomatology in the placebo group, and in BetaVia Complete. And you can see that for each of the 13 symptoms, BetaVia Complete was able to reduce them in the study. For example, we saw at 30 days a statistical significant reduction in tiredness and headache and body ache, and again at 90 days an effect on sore throat and headache. One interesting question is how early does BetaVia Complete start to work? Again, this question can be answered through the analysis of the WURS questionnaire that has to be fulfilled by participant every day from the beginning of the study. So we saw that BetaVia Complete group start to diverge from placebo within the first week of supplementation. Specifically, we saw that the URTI symptoms were reduced compared to placebo at 3 days after 3 days of supplementation, and this effect was sustained throughout the 90 days of supplementation. In terms of global severity, we saw a trending effect after 1 week supplementation and a statistical significant reduction compared to placebo from 2 weeks of supplementation on. And similar effect was observed for URTI symptoms, URTI episodes, also in this case, we saw a statistical significant reduction after 2 weeks of supplementation sustained throughout the study. So summarizing the significant findings from the a priori analysis, we saw that healthy participants taking BetaVia Complete for 90 days reported fewer sick days, symptoms, symptom days, episodes, and URTI, global severity of URTI symptoms. Also, in terms of early effect, we saw statistical significant effect compared to placebo at 3 days for URTI symptoms, after 2 weeks of supplementation for global severity and episodes, and at 30 days for fewer sick days. So the data presented indicate that BetaVia Complete can be a new and sustainable ally for year-round support, immune support, either in sport nutrition or activities indoor or outdoor, or in leisure time for the whole family. So to conclude this presentation, BetaVia Complete is a flexible ingredient for different applications. We collaborated with partners such as Truffini and Regge Farmaceutici in Italy or Edenkamp in Germany to develop a ready-made solution with BetaVia Complete in combination with vitamins and minerals. From the classic hard shell capsules to sustained release tablets, stick, or effervescent tablets to dissolve in water, BetaVia Complete can offer a different opportunity to innovate in the immune platform. Also, we developed gummies with a dose per gummy that make it suitable for the whole family, so different gummy for the different member. And also, BetaVia Complete has been included successfully in chews, in bars, in bites, and in fruit, vegetable, and chocolate drink mix. So I thank you for your attention, and Nick, I hand it over to you.
Thank you, Samantha, for that great presentation. We would like to inform our audience about the upcoming webinar on Kemin's Innovation Series on November 24th. With that, it is time to turn to the question and answer portion of today's webinar. Just to remind everyone, please feel free to type any questions you have into the Q&A box below. Our speakers would love to hear from you and answer any queries you may have. Our first question today is for Samantha. So Samantha, what is the regulatory status of BetaVia Complete in Europe?
Oh, thank you, Nick. Very important question. BetaVia Complete is already GRAS, say, due to the source of the product in Europe. BetaVia Complete falls under the scope of the Novel Food Regulation. And Kemin submitted the dossier in December 2018. So after reviewing the data presented, the production, the characterization of the ingredient, and also the safety data generated by Kemin and belonging to Kemin, EFSA has issued in May this year a positive opinion where the panel confirmed the safety of the wool algae fermentate, so BetaVia Complete, for the purpose use and use level. So the target group are the general population for food and beverages, and the general population above the 1 year of age for food supplement. And the Commission implementing regulation authorizing the place in the market of BetaVia Complete as discussed, and during the meeting in September this year, and all the member states had voted in favor to the approval of this regulation. And so we are now waiting for the publication of the regulation in the official journal of the European Union, and this will give Kemin a 5-year exclusivity on the commercialization of BetaVia Complete, and this should happen in the course of next weeks.
Okay, great. Thank you for talking us through that. Our next question has come in for Professor Calder. So during your presentation, Philip, you mentioned that beta-glucans prime the immune system. So what is the difference between priming and activation of the system?
Thank you. That's a good question. I think there's a subtle difference, and the immune system exists to be activated by and responsive to pathogens and pathogen-derived materials. So in the face of an infection, there's a huge activation of the immune system, which is designed to deal with the infection, and that's transient. Priming is a little bit different. So priming is really getting the immune system all ready to go if there happens to be an infection. And this is a really, I think, intriguing role of the beta-1,3 glucans, that they have this ability to, if you like, put the finger on the trigger of the immune response, but they don't actually pull the trigger. That requires the presence of the infective agent. So I think this is a subtle difference, but it's really getting the immune system, priming is getting the immune system all ready to go if there happens to be an infection. And of course, if there isn't any infection, the immune system doesn't need to become activated.
Okay, great. Thank you for that. We now have a question that's come in for Jeff. And the question, Jeff, is, are beta-1,3 glucans found in other algae?
Yeah, that's a great question, Nick. Actually, beta-1,3 glucan is pretty specific to Euglena. And so Euglena is a genus level. So there are other species of Euglena that can make beta-1,3 glucan. But the other types of algae that you might be familiar with, like spirulina or chlorella or other types of, I guess you'd call them regular algae, they don't have that ability. So Euglena is pretty unique in that regard.
Okay, thanks for that answer. And then Mike, there's a viewer that wants to know, what would you consider to be the key changes in consumer attitudes towards immunity with the current global situation?
Yeah, obviously, it's a sensitive topic. What we're seeing at the moment is when it comes to health, consumers can often have aspirational attitudes whereby they will want to improve their health, but they will tend to have a short-term focus on health. They'll tend to have specific health goals, and when they're either achieved or not achieved, consumers can sometimes slip back into bad habits. It's a key reason why lifestyle-related health problems continue to rise, despite consumers saying that they're attempting to lead healthier lifestyles. What we'll see as a result of 2020 and beyond is that this attitude behavior gap will somewhat diminish. And as a result of this, consumers will take a more proactive and long-term approach to health, because issues such as health are no longer aspirational, but consumers are more concerned about basic things such as disease management and immunity and their vulnerability to illness, and how certain aspects of their life are having a direct influence on these issues. So I think what we'll see is a lot more of a proactive approach. We'll see a back to basics approach to focusing on basic well-being as well. So, and I think it's something that will significantly influence the consumer over the next couple of years, especially as consumers think that the current situation is one that's likely to exist for potentially a couple of years.
Thanks for that, Mike. And we've now got another question that's come in for Professor Calder. So Philip, how should bioavailability be considered for beta-1,3 glucans?
Yeah, that's another good question. So I think it's really important to keep in mind the importance of the gut-associated immune system, which I talked a little bit about. And what's clear from the animal study that I mentioned is that immune cells from the gut wall can access beta-glucans from the gut lumen, and then they can process them. So what that means is actually you don't need a structure to be bioavailable to elicit the immune effects. And I think this could be another sort of advantage of beta-1,3 glucans in terms of their immune modulation, that it doesn't require systemic uptake. They're actually acting through the gut-associated immune system.
Okay, thank you for that. So we've now got another question that's come in for Samantha about your product, and the question is your product stable under high temperatures?
Well, another good question in light of formulation and application. The short answer is yes. So expanding a little bit on this, so we saw that the regulation authorizes BetaVia Complete for use in food and beverages, different kinds of food and beverages. And the basis for this decision has been the data generated by Kemin and provided by Kemin, in support that show that the beta-1,3 glucans in BetaVia Complete are able to be uptaken and taken up by, so recognized and taken up by the cells of the innate immune system after BetaVia Complete has been in autoclaving cycle for 30 minutes at 135 degrees and 30 psi. So really very harsh condition that confirm that the product is really stable under high temperature and can be used in different applications.
Thank you. We've now actually got another question for you, Samantha. We'll keep you on the line. So the question is, what would be the percentage of beta-glucan in the ingredient?
Okay, yes. This is, as Jeff was explaining, the algae store beta-1,3 glucan as a source of energy naturally, and is enabled to high concentration of the product that is naturally present, and then we just collect and dry the product. So the minimum percentage of beta-1,3 glucan in BetaVia Complete is 50%. And then in addition to this, being a whole algae fermentate, we have the other nutrients such as the protein and the mineral, the vitamins there.
Okay, thank you for that answer, Samantha, and for all of your contributions to this webinar. I'm afraid we're now out of time today. If we didn't get round to your question, we will soon send you an answer by email. I'd like to thank our speakers again for being with us today and to remind you at home that this webinar will be available on demand for the next 3 months. Thank you for being with us today, and goodbye.